Hello, good evening students. Welcome to GDC classes and another video lecture on medicinal chemistry. Hope you all are doing well. So without wasting any time, let's start with our today's lecture on adrenergic drugs. So what are adrenergic drugs? Adrenergic drugs are also called as sympathomimetic drugs. Sympathomimetic drugs. So what are sympathomimetic drugs? They basically mimic the action of sympathetic system. Okay, so sympathetic system. So what is sympathetic system? It basically prepares our body for fight and flight condition. So whenever our body faces the condition of stress, our adrenergic system gets activated, and due to activation of this adrenergic system, there are various changes in our body, like the heart rate increases, the sweating also increases. So why the sweating is increases? That it helps in maintaining the body temperature. So the sweating increases, the flow of blood towards our brain increases, as we need our brain to work actively during the stressful conditions. So the blood flow towards our brain also increases. Now as well as the kidney functions suppresses and the glands also suppresses, except the sweat gland, as we need to maintain our body temperature. Now there are three types of neurotransmitter that works for adrenergic system. Okay, so A and D. First is adrenaline, second is noradrenaline, and third is dopamine. So out of these three, noradrenaline is the prototype neurotransmitter. Okay, now moving forward, now if we classify these adrenergic drugs, they are basically divided based on the chemical moieties as the catecholamines and non-catecholamines. So what are catecholamines? The drug moieties or the chemical moieties that contain catechol ring in their structure, in their chemical structure, are known as catecholamine, and those do not contain a catechol ring in their chemical structure are known as non-catecholamines. So catecholamines are basically of two types, the endogenous and the exogenous. The endogenous catecholamines, they are naturally occurring or they are naturally synthesized in our body, and exogenous catecholamines, they are synthetic ones. So as they are synthetic, so they are synthesized outside our body, and whenever there is a need, so we intake them or we take them. Now endogenous catecholamines like noradrenaline, adrenaline and dopamine. Exogenous catecholamines like dobutamine, isoprenaline. Okay. Now non-catecholamines that do not contain a catechol ring in their structure. So tyramine, ephedrine, amphetamine, phenylephrine and salbutamol. So here this is the structure of a catechol. So it is just a aromatic ring with two hydroxy group attached at 1 and 2 position. Now this catecholamine, this catechol ring is the precursor for many drugs, many adrenergic drugs. Okay. Now when this catechol reacts with ethylamine, it forms catecholamine. Now this catecholamine is also called as phenylethylamine, as it contains a phenyl ring, an ethyl chain and an amine. So phenylethylamine. Now if we add an OH group to the beta carbon of this phenylethylamine or catecholamine, it is known as phenylethanolamine. So now this phenylethanolamine is the chemical backbone of many adrenergic drugs. Okay, so it is very important. Now, moving forward towards the synthesis of catecholamines. So, the precursor for the synthesis of catecholamines is the L-tyrosine. Now, this L-tyrosine, in the presence of an enzyme, tyrosine hydroxylase, it forms DOPA. Now, this DOPA further, in the presence of an enzyme, DOPA hydroxylate, it forms dopamine. Now, this dopamine is the first neurotransmitter that is naturally formed inside our body, synthesized inside our body. So, it is stored in the sympathetic nerve ending and the adrenal medulla. Now, this dopamine, in the presence of an enzyme, dopamine beta hydroxylase, it forms norepinephrine. Now, this norepinephrine is the second type of neurotransmitter that is naturally synthesized in our body, and it is also called as prototype 1. Okay. Now, this norepinephrine, in the presence of phenylethylamine and methyltransferase enzyme, it forms epinephrine. Now, this epinephrine is the third type of neurotransmitter that is naturally synthesized in our body. Now, let's see the structure of these catecholamines. So, these three catecholamines are the three neurotransmitters that are naturally synthesized in our body. First is dopamine. Now, all these three catecholamines, as they are catecholamines, so they will contain a catechol ring in their structure. So, here is the catechol ring. Now, in dopamine, we have a ethyl chain and an amino group. We do not have a beta hydroxycarbon in dopamine. Now, second is noradrenaline. In noradrenaline, again we have a catechol ring, an ethylene chain bridge, and an beta hydroxy group and an amine in noradrenaline. Now, in adrenaline, we have a catechol ring again, an ethylene bridge, and beta hydroxy group and amine and CH3 attached to the amine group, that is adrenaline. Basically, the structure of noradrenaline and adrenaline is almost similar, just the difference is this CH3 group that is present extra in the structure of adrenaline. Now, moving forward towards the adrenergic receptors. Basically, there are two types of adrenergic receptors present in our body. First is the alpha adrenergic receptor and the beta adrenergic receptor. So, they are further subdivided into the alpha 1 and alpha 2 adrenergic receptor and beta 1, beta 2 and beta 3 adrenergic receptors. Now, the SAR of phenylethylamine. So, why the SAR of phenylethylamine? As I already told you that this phenylethylamine is the chemical backbone. So, it is the chemical backbone for various adrenergic drugs and the naturally occurring neurotransmitters like the epinephrine, norepinephrine. Okay, so that's why this phenylethylamine is very important. So, this is the structure of basically the phenylethylamine. It has a very simple structure. So, this is the phenyl ring. Now, here we have an ethanol. Here we have amine group, and as it contains a catechol rings, we have two OH group. So, this is the structure of phenylethylamine. Okay, so if now talking about the SAR of this phenylethylamine, now substitution on phenyl ring. So, if we talk about only this phenyl ring, if we make changes to this phenyl ring, what effect will it give towards the adrenergic activity? So, whenever there is a direct acting adrenergic drug, it basically contains the hydroxy group at the meta and the para positions. So, here as we can see, so this is the ortho, this is the meta and this is the para. So, at meta and para position of this aromatic ring, we have a hydroxy group. That is important for a direct acting adrenergic drug. In direct acting adrenergic drugs, we have this, and we also have a beta hydroxy group. Beta hydroxy group is also present in direct acting adrenergic drugs like in norepinephrine and epinephrine. These are directly acting adrenergic drugs and contain a beta hydroxy group, and at meta and para position of aromatic ring they contain a hydroxy group. Now moving forward, now if we replace this catechol moiety, now as we know in catecholamines we have a catechol moiety. Now if we change this catechol moiety with resorcinol, so what is resorcinol? Resorcinol is just similar to catechol, just the position of OH changes here. So here we have at 1 and 3rd position we have the OH group. So if we substitute this catechol ring with resorcinol, then it will result in the increase in its beta 2 selectivity. Okay, and it also decreases the metabolism by 𝘾𝙀. Now what is this 𝘾𝙀? Now as if we are changing this catechol ring with resorcinol, then its metabolism by 𝘾𝙀 is decreasing. So 𝘾𝙀 is basically the catechol O methyl transferase. So what is this? This is basically the enzyme that is responsible for the metabolism of the catecholamine. So as in this terbutaline we have substituted the catechol ring with the resorcinol. So now this will not be able to metabolized by the help of this enzyme 𝘾𝙀. So the metabolism by 𝘾𝙀 also decreases by the replacement of catechol ring by resorcinol. But this norepinephrine and ephedrine can be metabolized with the help of this 𝘾𝙀. So this results in the increase in the duration of its action. Now moving forward, now if in this catechol ring at meta position the hydroxy group, if we change it with methanol group, so it will result in an increase in beta 2 selectivity. Beta 2 selectivity increases, and again the decrease in the metabolism by 𝘾𝙀. Why the decrease in the metabolism by 𝘾𝙀? As the 𝘾𝙀 is responsible for the metabolism of catecholamines. Now as we have made a change in this catechol ring, so now it is not anymore a catechol ring, so it will not be able to metabolized by 𝘾𝙀. Okay, now this is salbutamol. Now if this catechol ring, in catechol ring at para position, if we remove this hydroxy group, okay, so if we remove this hydroxy group at para position of epinephrine, then it will be selective towards the alpha-1 adrenergic receptor. Okay, now the name of this drug is phenylephrine. Now moving forward towards the substitution on the ethylene linkage. Now the phenylethanol amine, it also has a ethylene bridge. This is not there. So here it has a amine group and here we have a aromatic ring. So right now we are talking about this ethylene bridge. Now this ethylene bridge is very important or very important for the activity of the adrenergic drugs. Okay, like it is present in norepinephrine and epinephrine. So here is the ethylene bridge, here is also an ethylene bridge present. Now if we make a substitution in this ethylene bridge, substitution by a methyl group or a ethyl group. If we substitute this alpha position here with a methyl group or an ethyl group, so it will result in the decrease in the metabolism by 𝘂. Now what is this 𝘂? 𝘂 is also an enzyme, monoamino oxidase. So 𝘂. So 𝘂, this is responsible for the breakdown of monoamine. So as if we substitute a methyl or ethyl group at this alpha carbon, so its structure now is different from the natural monoamine. So as it is different from the natural monoamine, so MAO will not be able to recognize this structure and it will not degrade its structure now. So that's why the decrease in metabolism by MAO will take place for amphetamine. Okay, now moving forward, now as this hydroxy group in this epinephrine, now if we talk about the epinephrine, now in the epinephrine structure, this OH, the hydroxy position, the beta hydroxy position, the OH here is responsible for the chirality of this epinephrine. As now it has 4 different substitutions, like this is the first substitution, this is the second substitution, this H is the third substitution, and this H is the third substitution, and this OH is the fourth substitution. So this carbon here has 4 different substitutions attached. So now this carbon is a chiral carbon, and due to this OH only, this epinephrine shows chirality. Okay, now this chirality can be R form and S form, and this R form of enantiomer is more potent. How much more potent? 12 to 15 times more potent than this S epinephrine. Okay, now moving further towards the amino terminal. Now in phenylethynolamine, we are talking about the amino terminal. Now this amino terminal is also very important for the direct adrenergic agonistic activities, like seen in epinephrine and terbutaline. Okay, now this primary and secondary amine, if there is a presence of primary and secondary amine, then the structure is more potent, but if we change it with tertiary or quaternary amine, then its potency decreases. Okay, if we compare it. Now this amino substitution, now the substitution at this amino group results in the selectivity, change in the selectivity of the chemical. Okay, now if there is the substitution of bulky group with this amine, so it will result in the decrease in the alpha selectivity and increase in the beta selectivity. Okay, like here the isoprenaline is the example. Now here, as at NH2 or amine group, if we attach a bulky group, that is an isopropane group, so this isopropane group will result this isoprenaline to be beta selective. Okay, so due to the attachment of the bulky group to the amino group, its beta selectivity increases. Now the large substitution of amino group decreases the metabolism by MAO. So why the monoamino oxidase metabolism decreases? Same reason, as the structure differs from the natural monoamine, so the MAO is not able to recognize this structure. So as it is not able to recognize it, so it will not be able to degrade it. So terbutaline is is not metabolized by MAO, and its duration of action increases. Okay. Now moving forward towards the classification of adrenergic drugs based on the mechanism of action. Now first is direct acting agent. Direct acting agents means they act directly on the receptor of adrenergic drugs. Okay, so first are the selective alpha-1 selective drugs. Okay, alpha-1 adrenergic selective drugs. Now first is phenylephrine. So here we will basically study about how, as there are many drugs present in a particular class or particular, like in adrenergic drugs also there are many drugs. So it is very difficult to remember each and every drug structure. So here I'll tell you about some tricks by which you can remember the structure of these drugs easily. It will easy to remember for you. So first is phenylephrine. Okay. So by this structure is almost similar to epinephrine. Okay. But it lacks a catechol ring. So in epinephrine we have a catechol ring. What is catechol ring? It is basically a benzene ring with 2 OH. So in phenylephrine we have a phenyl ring. We have an OH at meta position, but at para position the OH is not there in phenylephrine. In epinephrine it's there. So phenyl, we have a phenyl ring, we have OH attached here. So and the rest of the structure is almost same as epinephrine. We have a ethylene bridge, we have a beta hydroxy group, we have NH, and we have CH3 here. So almost similar to epinephrine, just the difference is that at para position we do not have hydroxy groups here. So phenylephrine, easy to remember. Now this phenylephrine is resistant to COMT. Now why it is resistant to COMT? As COMT is only able to metabolize those drugs which have catechol ring. Now this, as at para position the OH is removed. So now this is not a catechol ring. So the COMT will not be able to metabolize this chemical structure, chemical drug. Okay. Now second is methoxamine. Now by the name we are getting to know methoxa or methoxy. So what is methoxy group? OCH3. It is a methoxy group. So its structure is almost similar to epinephrine, just the difference is that instead of OH now we have OCH3 at para position to each other. So ortho, meta, para, ortho, meta. At ortho and meta position now we have OCH3 group, that is methoxy group attached, and the rest of the structure is similar to the epinephrine. So it is used as a nasal decongestant. Okay. Now moving forward towards the drug that is alpha 2 selective. Okay. Now first is clonidine. Okay. So from clonidine, from Cl you can remember chlorine. Okay. So it is basically used in migraine and glaucoma. So clonidine ring is a imidazoline derivative. Okay. So it contains an imidazole ring, it contains an NH and a chlorine here. So clonidine. Now second is apraclonidine. So its structure is almost similar to clonidine, just the difference is that as we know A, so from A we can say azo. Azo means nitrogen, and from PRA. Okay. So aza. Ortho, meta, para. So this is ortho, meta, para. So A, azo, pra, para. So azo, that is NH2 at para position. So it is almost similar to clonidine, just we have to attach at para position, we have to attach an NH2 group. From NH2 we can remember from this A, that means azo, azo means N. So amine group is attached at the para position of this ring. Okay. So apraclonidine, easy to remember. Okay. Now third is brimonidine. Brimonidine, from here we can say Br. Br means bromine. So we have a bromine here. The other, it's same, the imidazole ring, it contains an imidazole ring. It is also used in glaucoma, So brimonidine. Bromine is here attached to this. Now fourth is dipivarfrin. Okay. So from dipivarfrin, it is basically a prodrug of epinephrine. As it is a prodrug of epinephrine, so its structure will be almost similar to epinephrine. So almost the structure is similar to epinephrine, just it is esterified with the 𝘂𝙑𝐿ic acid. So what is 𝘂𝙑𝐿ic acid? Now this CH3 with double bond OH. Now this is 𝘂𝙑𝐿ic acid. Now it is esterified with this 𝘂𝙑𝐿ic acid. When epinephrine, it is esterified with this 𝘂𝙑𝐿ic acid. Esterification means the removal of H2O. Then it will form this dipivarfrin. So here this group is attached both the sides. So this is the structure of dipivarfrin. It is almost similar to the structure of epinephrine. Okay, easy to remember. Now moving forward, the non-selective alpha agonist. So basically it can act on both alpha 1 and alpha 2. They are not selective for a particular receptor. So first is tolazolin. Okay, so from tola we can say toluene. So what is toluene? Toluene is basically a benzene structure with CH3. Okay, so here tola, toluene. Okay, and from azolin we can say imidazolin. From zolin we can say it has a imidazole. It is an imidazole derivative. So zolin imidazole ring. So tolazolin, it has a benzene ring with CH2 that is attached to imidazole ring. Okay, so it is an imidazole derivative that is used for the vasodilation. Easy to remember, tolazolin. Second is nefazolin. Now nefazolin, so here this also contain a imidazole ring, and this, it is very simple to remember. Okay, so nefazolin, almost similar to tolazolin, just this ring is different. Just this is from nepha, you can say naphthalene. Okay, so this is basically the naphthalene ring. So naphthalene ring attached to imidazole, nefazolin, imidazole ring. Okay, now third is oxymetazolin. Okay, so from oxy you can say hydroxy. Okay, from, so here we have imidazole ring, now we have oxy, we have hydroxy attached. Okay, so imidazole ring attached to some hydroxy, so oxymetazolin, almost we can remember it, oxymetazolin. It is also very simple. Now xylometazolin, from xylo we can remember xylene. So what is xylene? Xylene is basically a benzene ring with two methyl group attached. So here we have a xylene and then imidazole. So it is also a imidazole derivative. So imidazole ring with a xylene ring and a isopropane, butane. CH here. Okay. So here this is xylometazolin. Easy to remember. Okay. Now selective beta 2 agonist. Now these drugs are beta 2 agonist. They are selective towards the beta 2 receptor. Now first is terbutaline. Okay, so from buta we can say we have 4 carbons. Okay, so 1 2 3 4. So here we have butalin. Okay, so this terbutaline, we have a resorcinol in the place of catechol ring. Okay, almost the other things are same, like here we have a beta hydroxy group, here we have a ethylene chain, here we have amine, here we have we have 4 carbons, so buta, so here from here we can learn buta, and here we have a phenyl, we have a resorcinol. Okay, so terbutaline, as this is a resorcinol, so it will not be able to metabolize from form. Okay, resistant to comp. Now second is salbutamol. Now for salbutamol, from sal we can say salicylic acid. So this structure here is almost similar to salicylic acid. So in salicylic acid, here we have COOH. So in place of COOH, we have here CH2OH. So this structure is almost similar to salicylic acid. So from sal, we can say similar to salicylic acid. Buta, we again have 4 carbons here. Okay, all we have a OH group here, and the rest is ethylene chain and NH or amine group. Okay, the other things are almost same. So it is basically used in COPD or the lung disorders. Now pyrbuterol. Okay, so from pyrbuterol we again have a bute here. So bute, from pyr we can say pyridine. From pyr, pyridine. From bute we have a butane, so we have a pyridine ring here, we have a butane ring here. All, from all we can say we have OH here. Okay, so pyrbuterol, we can simply remember the structure of pyrbuterol. It is also used in the treatment of asthma. Okay, now non-selective adrenergic agonist. These are non-selective. They are not selective for a particular receptor. They can act on different receptors. It can act on alpha receptor, beta receptor, alpha 1 or alpha 2, beta 1, beta 2, anywhere. Okay. So isoproterenol. So first is isoproterenol. Okay. So isopro. So from isopro we can say isopropane. Okay. So here we have isopropane. Now here we have a catechol ring. Okay. Now here we have a beta hydroxy group. Here we have NH. Here we have ethylene. So this structure is almost same as the other catecholamines. Okay. Just the difference is that here we have isopropane ring. Okay. Isopropane chain attached to the amine. Okay. Now second is epinephrine. Now for epinephrine, this is also called as adrenaline, and it is a natural catecholamine. The structure of this we have already discussed, but again we can discuss. So here this is the catechol ring. Okay, at beta position we have a hydroxy group, NH here, and we have a CH3 here. So this is epinephrine, a very simple structure. Now third, norepinephrine. In norepinephrine we just have one difference, that we don't have CH3 here. Here we have NH2. The rest is same. We have a catechol ring, a beta hydroxy group, and an ethylene chain. Now moving forward, indirectly acting agents. So these do not directly act on the receptor of the adrenergic drugs, but they act by the release of the endogenous catecholamine. So what are endogenous catecholamine? Like the adrenaline, noradrenaline. So these are the endogenous catecholamines. Okay. So first is amphetamine. So amphetamine, its structure is very simple. We just have a benzene ring, an amine, an ethylene chain, and a CH3 here. We already discussed this before. Due to this CH3, the 𝘂 enzyme do not act here or is not able to degrade this monoamine. Remember, we have already discussed this. Okay, so it basically stimulate or synthetic stimulant of CNS that increases the activity of dopamine and norepinephrine. So this dopamine and norepinephrine, they are basically the endogenous catecholamines, amphetamine. Okay, so it's a very simple structure, amphetamine. So hope you understood the video and liked it. Thank you so much.